In this lecture, we will discuss the metabolism of histidine, which involves both biosynthesis of histidine and degradation of histidine. First, we will deal with the biosynthesis of histidine, which involves the participation of PRPP and ATP in the formation of N' 5' phosphoribosyl ATP. 5 carbon atoms of PRPP, 5 phosphoribosyl alpha pyrophosphate, and 1 carbon of ATP participate in the formation of N' 5' phosphoribosyl ATP. In this, a molecule of PPI, pyrophosphate of PRPP, is released out in the presence of the enzyme phosphoribosyl transferase, and the remaining atoms of the ATP which are not incorporated into this reaction are released or eliminated as 5-amino-imidazole 4-carboxamide ribonucleotide. N' 5' phosphoribosyl ATP in the presence of pyrophosphate hydrolase releases PPI of ATP and forms N' 5' phosphoribosyl AMP. N' 5' phosphoribosyl AMP in the presence of phosphoribosyl AMP cyclohydrolase takes in a water molecule and breaks the purine ring and decyclizes it and forms N' 5' phosphoribosyl 5-amino-imidazole 4-carboxamide ribonucleotide. This in the presence of isomerase enzyme forms N' 5' phosphoribosyl 5-amino-imidazole 4-carboxamide ribonucleotide. This in the presence of the enzyme synthase glutamine amidotransferase deaminates glutamine to glutamate. This reaction also releases 5-amino-imidazole 5-carboxamide ribonucleotide, which further participates in purine biosynthesis. So coming back to our original reaction, N' 5' phosphoribosyl 5-amino-imidazole 4-carboxamide ribonucleotide gets converted to imidazole glycerol phosphate. This imidazole glycerol phosphate in the presence of dehydratase forms imidazole acetal phosphate. This acetal phosphate in the presence of histidinol phosphate aminotransferase forms L-histidinol phosphate. Here, a molecule of glutamate gets converted to alpha ketoglutarate, and this is an irreversible reaction. Both the ways it is possible. L-histidinol in the presence of histidinol phosphate phosphatase forms L-histidinol. L-histidinol in the presence of histidinol dehydrogenase forms histidine. This phosphatase helps in the removal of phosphate from L-histidinol phosphate, and here in this step NAD gets reduced to NADH by forming histidine. Now let us see the degradation of histidine. Arginine, glutamine, histidine and proline are all degraded to glutamate, which in turn is oxidized to alpha ketoglutarate. Histidine's conversion to glutamate is more complicated. Firstly, histidine is non-oxidatively deaminated, then hydrated, and later the imidazole ring is cleaved to form an 4-aminoglutamate. The 4-amino group is then transferred to tetrahydrofolate. Let us see the steps in detail. Histidine in the presence of histidine ammonia lyase cleaves the ammonia group and forms urocanate. Urocanate takes in a water molecule and in the presence of urocanate hydratase forms 4-imidazolone 5-propionate. This again takes in the water molecule, and in the presence of imidazolone propionase cleaves the ring and forms N-formimino-glutarate. N-formimino-glutarate in the presence of glutamate formiminotransferase um, converts tetrahydrofolate to N-formimino tetrahydrofolate, thereby forming glutamate. Glutamate in the presence of glutamate dehydrogenase uh, reduces NADPH to NADP to NADPH and removes a molecule of ammonia by forming alpha ketoglutarate, which is a intermediate product of TCA cycle. Thank you.